Greetings everyone and welcome back to the third and last part of our talk. And this time we will apply what we have learned in the previous parts to answer the last question, what test to be used. Let's start our talk with this question. Imagine you are planning for new research. At which stage you will be able to select your test? Can you do it before or just after conducting your research? To answer this, let's go back to our 5 criteria for statistical test selection, where we will first determine our variables type, followed by our hypothesis type, and then we will determine the dependency of our observations, the number of variables we are dealing with, and lastly, the normality of our data. And if you remember, for the first 4 criteria, we can determine them from the aim, and sometime we might need to read the methodology of the study, and both are actually a part of our proposal, so can be determined before conducting the study. The only issue we will have with the normality of our data, where we actually need our data to run a normality test. And to be able to write our statistical analysis part in the proposal and select our test before even conducting the data, that can be simply done by selecting one parametric test and other non-parametric test and stating in the proposal that if our data were normally distributed, we will use this test, and if it's non-normally distributed, we will use its non-parametric counterpart, and by that we can determine our test even before conducting the research. Let's apply now and answer the question what. I prepare these two flowcharts to serve the purpose. The first flowchart is the parametric test flowchart, and you will use it only when your data is normally distributed. You will apply the 5 criteria, so you will first determine your variables type, starting with independent, followed by the outcome variables. And for the hypothesis, you will be actually guided by the variables type, as the hypothesis of association will take the right side of this chart, and you will be dealing with either regression or correlation test, while the hypothesis of difference will take the rest of the flowchart. After that, for the hypothesis of difference, you will determine your observation, whether it's paired or not paired, and how many variables you are dealing with, and lastly, you will be able to select your test. The second flowchart have the same rule, but it is designated specifically for non-normally distributed data, and that's why it's called non-parametric test flowchart. Let's take an example from literature to make things clear. This study comparing foreign intracanal medicament and addressing both the antibiofilm and the pH changes over time. So looking at the table, they are comparing 4 groups, or actually 5 groups, together with the control, aiming to measure the biofilm percentage of the live bacteria. The authors state that for this part, normality was absent, so we will go to our non-parametric test flowchart, and our predictor variables are the 4 or 5 groups, so they are categorical. Our outcome variable is the biofilm percentage, so our dependent variable is quantitative, and each group is different from others, so our variables are unpaired, and they are more than 2, so Kruskal Wallis should be selected, and that's actually what the authors did. But if you notice, they used additional test beside Kruskal Wallis, which is a done test, and I will come to this in seconds. For the second dependent variables in this study, which is the pH, actually these authors target only to compare the 4 different groups to others. There is no comparison carried out within one group at different time interval. So again, here we are dealing with unpaired data. They state that their pH data was normally distributed, and this time we will go to the parametric test flowchart. Again, categorical independent variable, quantitative dependent variable, unpaired data more than two groups, and this time ANOVA should be selected since we are dealing with parametric tests, and that's what the authors did. But again, they use another test, which is Toki test, beside ANOVA. To clarify why Dan test and Tukey test were used here, put in mind whenever you compare more than two groups with ANOVA or Kruskal-Wallis, they will tell you whether there is difference or not, but they will not determine whether the difference is, for example, between group 1 or 2, or it's between group 2 and 3. And to know that, there is a specific type of test known as post hoc test, which should be applied after any multiple comparison. And these tests will be differ whether your data is normally distributed or not, or if your observation is paired or not. With Toki, Benferroni, Wilcoxon, and Dannett tests are among the most commonly used tests. So let's take one last example to further clarify this point, and that will lead us to the last point of our talk. If you take a look at the figures, you will be able to identify 4 of the criteria, and for the 5th criteria, the authors mention that the normality were absent. So this time we will go to our non-parametric test flowchart. We are comparing, as we see in the figure, 4 types of intracanal medicaments. So our predictor variable are categorical, and we are aiming to measure the CFU. So it is a quantitative dependent variables. For our observation type, their aim first was to compare the 4 groups to each other, so our data is unpaired and there are more than 2 groups, so they should select Kruskal-Wallis test. As a secondary aim, they want to compare within one group, for example, the ibuprofen, the CFU before applying the ibuprofen and after applying the ibuprofen. So here we are dealing with bare data and 2 variables, one before and the other after within the same group. So Wilcoxon should be used, and that's actually what the author did. But if you notice, they selected third test, which is a Mann-Whitney test. A Mann-Whitney test, if you go back to the chart, is specifically designed for comparing 2 variables for unpaired data. And that's actually an error. And I mentioned this article at the very beginning of our first part, if you remember, when I said that by paying attention to this presentation, I was able to identify errors. So the first common error in statistical test selection is to use t-test and Mann-Whitney test for multiple group comparison. What the author fell in is that he, instead of comparing the 3 groups and then running a post hoc test, they did a separate comparison between each 2 group, and that's absolutely a mistake. He should run a done test, not a Mann-Whitney test here. As Mann-Whitney test, there is no attempt to control the type 1 error that done test will do. Other common error that this article fell in this article, there was actually no specific statistical analysis part. They just write these 2 sentences at the end of each methodology. And if you notice, the author run immediately ANOVA test. They didn't mention anything about normality test. So it's either they don't run a normality test or they intentionally want to use parametric tests because parametric tests are stronger and the inferences made from parametric tests are capable of generalization to the entire population, while those for non-parametric tests cannot be generalized. So the error is to use a parametric test for non-normality distribution or simply not to run a normality test. The third and last error is when dealing with ordinal data. For example, our periapical index when dealing with scores. The authors here deal with the score as an actual number and the frequency of occurrence of such score as mean and run a parametric test, and that's absolutely wrong. Actually, the other name for non-parametric test is the ordinal test, and therefore these tests are specific for such situation. And with scores, you don't have to run a normal test. You should immediately use a non-parametric test. By this, we can end our statistical journey. I hope you find it beneficial, and by the end, you was able to appreciate to which extent that topic is important for us as a researcher, and you will be able to identify errors and no longer skip the statistical analysis part of the scientific publication that you read. Thank you for listening and have a nice day.